All right, welcome again, everyone, to the ARPA-H Small Business Program information session. We're going to be getting started. We are thrilled to have you with us today and really look forward to sharing more information about the Small Business Program, including how to submit an application, eligibility considerations, and details of current open topics. My name is Jordan Ashwood, and I'm a Senior Program Officer with the ARPA-H Investor Catalyst Hub, managed by VentureWell. And we at the Investor Catalyst Hub are very excited to be hosting this webinar today. So before we jump into today's session, I did want to share just a brief overview about the Investor Catalyst Hub. So we at the Investor Catalyst Hub, it's a health innovation ecosystem connecting 300 plus members in 47 states across a fragmented health ecosystem. We leverage a hub and spoke model. So Investor Catalyst Hub spoke members have access to a variety of opportunities, including the ability to engage with ARPA-H program managers, attend IC Hub member-only events, access research and development opportunities, connect with other spoke members and really benefit from their unique capabilities and areas of expertise, and provide feedback on ARPA-H efforts through network surveys. And really, as a whole, the Investor Catalyst Hub's goal is to connect researchers, entrepreneurs, investors to accelerate the transition of innovative ideas into accessible solutions. And we do so by providing support in a number of different areas, including analyzing the commercial landscape, navigating regulatory pathways, and developing sustainable business models. So we welcome you to follow us on our social media, which has been shared in the chat, to just stay up to date on current events and opportunities, including applying to become a spoke in our network. So with that, I will now pass it over to Jenica Patterson, Patio Division Director.
Hi, everyone. Welcome. I'm Jenica Patterson. I am one of the portfolio leads in our transition office, and I run the small business program for ARPA-H. And I'm so excited to see the numbers here. We have close to 400 people participating. We are so excited to see such excitement and interest in the small business program at ARPA-H. So thank you for participating today. I want to give a special shout out to our Investor Catalyst Hub for hosting this event and spreading the word through the community. And also, I want to also give a shout out to GSA, who is running our contracting shop for our small business program for this solicitation. So thank you so much. And I'm going to first talk a little bit about who we are, who is ARPA-H, why are we so unique, and what our small business program is all about. Next slide. So if you haven't heard anything about ARPA-H, I'd like to start with our mission, which is very important. We want to make sure that we are accelerating better health outcomes for everyone. And we do that in multiple ways through our funding announcements, connecting with people through our Investor Catalyst Hub and our Customer Experience Hub located in Dallas, Texas. And we really try to bring in innovations that will disrupt the health ecosystem. And I'm going to talk a little bit about how we do that. If you go to the next slide. So ARPA-H, we are solely a funding agency. We were launched a little over 2 years ago. So we are what I call the toddlers of the federal government. We have been building our agency from scratch, including the small business program, since 2 years ago. So it's been really exciting opportunity to build a new federal agency. Again, we are solely a funding agency. Many people know we are connected to NIH, but we are an independent component of HHS that is housed within NIH. The reason we were able to stand up so quickly is because we were able to use some of the infrastructure that already was built within NIH, which was great. We are so grateful for. And we have wonderful collaborators across all the institutes and centers at NIH. We are not an institute. We are our own federal agency, and we do not do any internal intramural research. So we have no research labs. We put out funding announcements like we did for the small business program to make sure that we are bringing in proposals for people to do the work on the outside. We have approximately 2.5 appropriations for our budget. And again, it's independent from NIH. And we are very hands-on here at ARPA-H. So we do not fund grants. We typically fund contracts, cooperative agreements, and other transactions. And as you'll learn today through GSA and their particular part of this conversation, they'll talk about our contracts and what we're doing there. We have our program managers that have a lot of active management, hands-on management with our awardees to make sure we're meeting milestones and we're meeting that mission statement that I just mentioned in the previous slide. We are a bottoms-up, problem-centric approach organization. So what I mean by that is we don't bring in the ideas from the top, from our director's office. We hire program managers that actually come in with really unique creative ideas, and they are the ones that launch large programs. They come up with the topics for our small business program, as an example, to be able to build out their portfolio and be able to accelerate health outcomes for everyone. We are very lean and nimble. We try to work very fast and efficient. So we have really unique structures and legal authorities within our organization to be able to do that. Our director, she reports directly to the HHS secretary. We also have term limits to our program managers. So they come in with that really creative idea. They are here for 3 to 6 years to launch that idea and make it a reality, and really to help meet our mission. And we have a lot of flexibility in hiring too, to make sure we're bringing the brightest and the best program managers we can to be able to meet our mission. And we are a problems-focused organization. And you'll see that today, especially when you read through our solicitations and the small business solicitation. We come with that problem, but we expect people on the outside to be able to bring that solution to us so we can understand, how are you going to solve that problem for us, and how are you going to make it a reality? If you can go to the next slide. So to be able to meet our mission at ARPA-H, we have 4 or 5 mission areas, or 5 technical offices across the agency to be able to do that. Within our solicitation today in the small business program, 4 of the 5 here are represented across the agency, which is really exciting. We have 4 mission offices that propose topics that were placed into a solicitation. I work in the transition office, also known as PATIO, the Project Accelerated Transition and Innovation Office. We run the small business program, and we work collaboratively across our mission offices at ARPA-H to be able to come up with those unique ideas to foster innovation in the small business space. I'm not going to go through all of the mission offices, but I encourage you to look at our website, our arpah.gov website. If maybe these topics don't fit within your area, or maybe you have another unique idea that you want to present to ARPA-H, there are a lot of other opportunities to get funding through ARPA-H. There's through our programs, like I mentioned, the program managers launch these really large program initiatives. We also have open broad agency announcements, also called ISOs, for each of 4 mission offices that are highlighted in gray here. And if you have an idea that you think aligns with that particular office, you should submit your idea. It's a really easy 3-page abstract that you can submit, and they can let you know if it fits within their mission and to submit a full proposal, or it does not fit within the mission. So that's another really great way to work with ARPA-H. You also can be a spoke in our Investor Catalyst Hub, who's helping with the webinar today, as well as our Customer Experience Hub. So please take a look at all the resources we have online. There's a lot of great ways to interact with ARPA-H. Next slide. Today, we're here to talk about the small business program, though. And I want to say, I'm so excited all of you here today get the opportunity to be part of our first webinar ever for the small business program. So that's really exciting for ARPA-H. This is our 3rd solicitation for our small business program. So we keep growing. This is the biggest number of topics we've had. So right now we have 8 topics in our solicitation. So we're really excited to be here to present that to you today. So what does the small business program mean to us? Well, we really want to make sure we are leveraging research advances for real world impact. And we want to make sure that those are program manager led. So again, our program managers are the ones that generate these topics, and they typically align with their programs or their portfolios that they're looking to build within the 3 to 6 years that they are here. We make sure that we are launching at least 1 SBIR and STTR solicitation per year. This is our 2nd one this year, so we're really excited about that. And we also want to make sure that our small businesses are positioned for success that we fund. So we have a lot of opportunity to work with our small businesses that we fund, bring in some commercialization expertise from the outside, which I'll talk about a little more, to make sure we set them up for success. We're excited because we have a really simplified SBIR STTR application process. GSA is going to talk a little bit more about what that looks like. Really easy application process. And we try to expedite our review process. We want to make sure this solicitation, we are funding our awards by the end of this fiscal year. So that means around September timeframe. So right now it's July, we have August, September, we have 3 months to make that decision. So we work very fast. We, as many of you know, if you're small businesses, we do have stable and predictable non-dilutive funding. We are a non-dilutive funding agency. And we also want to make sure that we are meeting our mission to accelerate better health outcomes for everyone. And the way that we can do that and to get these innovations out to people is to make sure that IP rights are retained by the small business. So we really try to limit the amount that we're interacting in that space. And we also provide support through some of our programs that we've launched through our small business program to be able to help support what that IP process looks like. And again, we have a suite of entrepreneurial services that I'll talk about in a minute to help advance and commercialize these innovations. Next slide. And like I said, we do have an active small business program. I can't believe it. About a little bit over a year ago was our first solicitation. We had 2 program managers that existed at ARPA-H. We now have 18. But with our first solicitation back, I believe it was in April or May of last year, we were able to fund our first awards across 2 offices that existed at ARPA-H. And these here were the 5 topics, and just showing not only the diversity in terms of the innovations that we were looking for. You can see things all the way from digital health to minimally invasive surgery, but also the geographic diversity of how we awarded. We're excited as we build our program, we're going to build out that geographic diversity across the United States, as well as build out our diversity in terms of the projects that we fund. Next slide. So just some information about SBIR and STTR. It is one of the largest sources of early stage financing across the federal government. It is a congressionally mandated program. So if you spend, I believe it's more than $1 billion in appropriations, you are required to have an SBIR and an STTR program. And we are that. So we do both have an SBIR and STTR set aside. This is separate from the allocation for program budget for program managers. So this is a unique set aside that we have to make sure that we are fostering innovations in the small business space. For our SBIRs, we have a 3.2% set aside. And for STTR, we have 0.45% set aside of our entire budget, R&D budget for each fiscal year. Next slide. So who is eligible to apply for SBIR, STTR funding? And this is pretty typical across the federal government. So if you're used to getting funding through SBIR, STTR programs, we follow SBA guidelines. So I would recommend you taking a look at our website, but also taking a look at the SBA website for eligibility. But we are funding solely US businesses with 500 or fewer employees. And the PI that are primarily employed with the small business at the time of award and duration of the project. And we want to make sure for the SBIR and STTRs that more than 50% are owned and controlled by citizens or permanent residents of the United States. Next slide. The SBIR STTR program is a 3-phase program that's across the federal government. So it starts with phase 1, that's looking at early stage R&D efforts. So looking maybe at what is the realm of possibility for this idea to turn into a proof of concept. So that's really what phase 1 is. And typically the budget for phase 1 is a little bit over $300,000, funds for 6 months to a year. We are excited to say we have a budget waiver that exceeds that amount to $600,000 for 6 months to a year. For phase 2, that is a little bit of a later stage R&D effort. So looking really at what can we de-risk a little bit more, and what can we really focus on to help bridge that valley of death for commercialization. The budget guideline for phase 2 is about a little over $2 million for 1 to 3 years. We are excited to say that we have a budget waiver for $3.5 million for 1 to 3 years. So a bit above that cap. Phase 3 is really focusing on deep commercialization. You really have de-risked your prototype. You're ready to move out into the market. We aren't necessarily the customer. Our customer is all Americans. So we typically are not focused in that area for now, at least for this solicitation. We're focused solely on phase 1 and phase 2. If you recall, if you remember, I said we were part of NIH. So one unique authority we have is that we can do direct phase 2 funding. So what that means is typically you have to go from phase 1 to phase 2. We actually can go directly and fund you at phase 2. So you'll see within the solicitation that some of the program managers' topics say only phase 1 or only phase 2. You can identify where your technology fits there. And we also have an opportunity to do fast track. So that's a combination of doing a phase 1 and a phase 2 proposal, so you don't have a gap in funding between the two. Next slide. So there are some critical differences between SBIR and STTR. The biggest difference, I would say, is the partnering requirement. The STTR does require that the small business does have a nonprofit research institution partner. That could be a university, a nonprofit research organization, as an example. So that's the main difference between the SBIR and STTR awards, and if you're eligible. And another piece is the work requirement. So for the STTR, a big difference here is the minimum work requirements is 40% has to be the small business, 30% has to be the research institution partner. And the contract is always awarded to the small business. So for example, STTR, if the small business is working with a nonprofit research organization, we would be contracting to the small business with the sub-awardee as the STTR. Next slide. So like I said before, we're really trying to work hard to make sure the people that we fund, the organizations that we fund through the small business program, are successful. So we have established a public-private partnership with BioHealth Innovations. They are a nonprofit organization to bring in experts outside to understand how can we provide commercialization advice, expertise to our small businesses, so we can have user adoption as well as successful adoption into the market for our small business innovations. So we have a couple different areas where we do that. The Entrepreneur in Residence program is where BioHealth Innovation is supporting. So they have very well-established experts that can provide experience in business development, IP, user adoption, manufacturing experience, as an example. We typically pair people up with an EIR in phase 2 awards. We also have the ability to do needs assessment. So we do that through another public-private partnership through the Research Triangle Institute, RTI, and that's typically for phase 1. We want to make sure that we are understanding what are the needs of that small business. So the RTI provides an informal analysis of technical and business areas that are needed to be able to understand how that innovation can be competitive in the marketplace. We also know a big barrier to success is regulatory and reimbursement expertise. So we make sure that a lot of these EIRs bring in advice for navigating the FDA regulatory process, as well as reimbursement strategies to make sure that your innovation you're creating is affordable and accessible to all. Next slide. We already have an active EIR program with those awardees that I mentioned earlier. And exciting to say it's been pretty successful so far. I think the big piece to point out here is our EIR program has really helped companies reach inflection points in their progress throughout their award. So for example, connecting with the FDA or breaking down and barriers for IP issues as an example. So there's a lot of opportunities for us to help small businesses reach inflection points otherwise they couldn't do without ARPA-H funding. Next slide. So take out your cell phones, take a look at this QR code. This is our request for proposals that's out on the street right now. And you can take a look at all of the 8 topics that we have there to be able to write your best proposal. Next slide. And so just quickly, and Kimberly, you're going to be on deck soon to speak, but we have some key timelines and for you to take a look at. Right now, we released our full solicitation, I believe, last week, so June 26th. The deadline for proposal submission, and this is the most important thing to remember, is July 26th, 2024 of this year. So that's less than 1 month from now at 11 a.m. Eastern time. And we'll talk a little bit more about that later on in our in the webinar. So stay tuned. But remember, July 26th, you need to have at 11 a.m. you need to have your submission submitted. Next slide. Again, we have some stellar program managers across our agency, across those 4 mission offices that will present now about each of their topics. So listen really carefully for what they're looking for, as well as what they're not looking for. And I'm going to hand it over now to Dr. Kimberly Steele, Program Manager from the Health Science Futures Office, that's going to talk about her topic. Kimberly, over to you.
Thanks, Jenica, and apologies for jumping in too quickly. Hi, everyone. As Jenica mentioned, I'm a program manager in the Health Science Futures Mission Office, and our portfolio, or my portfolio right now, is all things lymphatics. And so for this solicitation, we are looking at topic number 1, ambulatory edema monitor. Why? The problem really is that currently, and sadly, in 2024, physicians do not have proper monitoring devices to monitor for edema, whether it's in your extremities, your upper or lower extremities. What do we have? We have a tape measure, we have a finger pushing into a leg, and we have some technology, but it's not really accessible to everyone. So what we're looking for is something that we can get out to patients early in their homes so that doctors can actually detect subclinical edema or much sooner the edema in the extremities, in the body, before it becomes unable to prevent hospitalizations. And so right now, this has become a real critical barrier for docs because we can't detect it. What do we want? What solution? Well, we're looking for you to help us create wearable technology and on-skin interfaces. What could that look like? Stockings, patches, and we'd like it to monitor peripheral edema in a way that enables continuous assessment of fluid status, and preferably to be able to do it at home. To do this solution, we've created 3 technical areas. Technical area 1 is hardware censoring platform. And we are looking for specific metrics, in this case, sensitivity and specificity of at least 90% in comparison to a gold standard. In technical area 2, we're looking for a mobile application for this data visualization. And here, let's get our patient satisfaction up. We know what it's like to wear wearables, sometimes very uncomfortable. So let's get the patient satisfaction to greater than 85%. And you can come in and propose what that valid score is. And then technical area 3, web server with database for processing and storing data. It would be ideal if you come in with a targeted product profile and the metrics that you are going to minimally achieve and what you ideally would like to target. What's in scope? Well, this is open, as Jenica said, to SBIRs and STTRs. We'd like to have you work on novel development of these sensors and monitors that can bring transformative improvements and detect with good sensitivity edema in all the way to subclinical, so that we as doctors can act on it in a much faster fashion and prevent hospitalizations and poor outcomes. Also, we'd love if you could work on advanced or bring in advanced or innovative AI and machine learning to the existing technologies, and that hopefully will improve our prediction and diagnosis. What is not in scope are incremental improvements to already existing technology. Bring on your novel technologies for us. And with that, I'm going to pass it over to Maxim, and I'm going to get it wrong, but Stetanov, who is did I do it wrong?
Yes, but that's okay.
Okay, well, and
Okay.
Maxim is standing in for Paul Sheehan.
Right. Hi, everybody. My name is Max Tverovat, and I am standing in for Paul Sheehan, who is the program manager for this. So the problem they're trying to address is about 5.5 million kids and adults in the United States have some sort of cognitive disability. And we're specifically looking at autism and Down syndrome as being the most common. And those cognitive disabilities often come with communication deficiency or difficulties in learning how to use language. One of the most common tools that's used either in special education classrooms or for adults with cognitive disability is something called an augmented or assisted communication tool, so AACs. And they take shape of anything from little pieces of papers that you Velcro to a board with pictures to iPad applications or even specialized hardware, kind of as a bigger version of a speak and spell. The problem with those kind of devices right now is that they're limited to a vocabulary size of about 200 words. To compare, vocabulary size of a normally developing preschooler is about 3,000 to 5,000. And by the time they enter middle school, it's about 10,000. And most adults can use between 25 and 50,000 words. So with that level of limitation, those devices are generally limited to pragmatics. So food, bathroom, clothing, etc. And with that limitation, they are unable to communicate emotions and they're unable to be expressive with their language. So in a way, those assistive devices at the same time are helping them at the low end, but also holding them back in language development. So what we are looking at creating right now is an intelligent augmented assisted communication system that uses large language models to create a predictive and adaptive AAC platform that is usable in real-world settings. The hardest part of this work will actually be creating a user experience that is acceptable and interesting and engaging for both kids and adults with cognitive disabilities, and that adapts to their ability to use language and to their cognitive ability at, basically meets the kids where they are. One of the biggest other goals here is developing a system that works in multiple settings, not just in school, but lets especially adults take public transportation, hold down a job, and be really productive and fulfilled member of society. Too many adults with autism end up unemployed, especially if you're looking at people of color, a disproportionate amount ends up in jail or homeless. So we really need those kind of tools at the point where the education system drops them off once they turn 18 or so. So what we are looking to see is a system that works on commodity hardware that can be publicly released, and at phase one gives us an effective vocabulary size of about 2,000 words, so about level of typically developing 5-year-old. And by the end of phase two, more than 10,000 words, so really approaching adult level language comprehension. And I'm very interested in having things work on commodity hardware and not requiring any kind of specially developing hardware. One of the reasons for that is that it's extremely difficult to develop hardware and bring it to market in the scope and budget of this kind of program. So ideally, this would be a fully software solution utilizing tools that can be bought on the open market. And the second reason, of course, is the cost to the users. So it should be affordable to the users. We are asking for fast track proposals, so encapsulating both phase one and phase two. The reason we're not going direct to phase two is because we've set up a ladder. There's a number of steps and gates that will help you achieve the results. We're not trying to boil the ocean all at once, but we can start small and develop from there. So novel applications of predictive language models to augment as adaptive communication tools, novel UX ideas that will improve expressive capabilities of AAC tools. What's out of scope? Incremental improvements to AAC tools. And this is something that I am very adamant about, and we added this to the program. Everything developed here should be done in close participation with disability advocates. And I believe that's actually the first in history of SBIRs. We are asking the teams to include team members with autism or cognitive disabilities. We know that people with autism can be extremely productive in software development. That's been established. Bill Gates, Elon Musk, etc. They should be, as a goal of advancing equity and equality for people with autism and other cognitive disabilities, they should be included as a part of the team. So we are asking for teams to reach out to the community, reach out to the advocacy groups, and build teams that are fully inclusive and that are providing opportunities for community members to be innovative and to be part of this project. So with that, passing it on to John to speak about the next topics.
John, you are still muted.
There we go. That's much better. So thank you, Max, for the introduction. My name is John Sheehan. I am with the Scalable Solutions Mission Office in ARPA-H. I'm going to tell you a little bit about the topic we've got here on behalf of our office. So what we're really looking for here and kind of some of the background on the thinking is that distributed manufacturing is something that we've been talking about for several years in the cell therapy manufacturing space. And I think there's opportunities in recent federal funding looking at genetic medicine manufacturing as well. So thinking about mRNA vaccines and other oligonucleotide-based therapies. So the initial inception of this small business topic was around what types of additional innovations in process analytical technologies or sort of rapid automated analytical product quality testing would be necessary to truly enable that distributed manufacturing of either cell therapy-based products or oligonucleotide-based products. So with that, we started to bucket this into a couple of goal areas. So starting off initially with the advanced inline or at-line process analytical technologies that could be used for either cell therapies or oligonucleotide-based therapies. So with the cell therapy side, we're thinking about soft sensors. This could enable certain chemometric type models to better understand the architecture of the metabolomics or other things that are ongoing in the cell. Or this could be actual cell biophysical measurements or viability measurements. So thinking about cell density, viability, or other biophysical properties. So really anything ranging from omics level data all the way towards information about the cell health and the cell population within the biomanufacturing process would be very useful to have very innovative process analytical technologies. On the oligonucleotide-based side, we're looking for process analytical technologies that can measure things like enzyme consumption, the concentration of nucleotide triphosphates, capping reagent efficiency, or the presence of things like process intermediates. So this could be, for example, double-stranded RNA that's becoming present during your in vitro transcription. So we really do want to look again at attribute-relevant process analytical technologies, things that could potentially inform the quality of the final product and help us to enable a more rapid release of these products should we get to that ultimate desired state of distributed manufacturing. But keeping in mind with all of this that while the goal is to try to have this towards a distributed manufacturing paradigm, these technologies as they're developed would equally be useful to centralized manufacturing paradigm. So keeping that in mind when we're developing these technologies, that they certainly could be dual use, but that we do have this vision of one day being able to do this in a much closer to the patient space. So moving on to the second goal area, the rapid and automated product quality testing. Once again, we're thinking here about sort of walk-up, very automated, very hands-off, if you will, quality control-based analytical methods. So again, we can think about analytical methods for both the cell therapy type manufacturing as well as looking at oligonucleotide-based products. But what I want to really try to emphasize here is that in order to do release in sort of this distributed manufacturing paradigm, we're going to have very simple to operate analytical equipment. So the user interface here is going to have to be very intuitive, be able to use by someone that maybe isn't an expert in that analytical technology. They could have, of course, expert support externally, but we want them to be able to walk up to the instrument, have an analytical method sort of preordained, and a very simple user experience to putting buffers or whatever other reagents need to go onto that equipment very quickly so that they can run the analytical methods very seamlessly. So we would like to have sample acquisition all the way to results in less than 24 hours. Again, we're looking for very rapid turnaround time of these analytical methods. And again, this software should really be enabled to have very clear pass-fail metrics to enable that user to see my product is meeting the required specification or it's not meeting the required specification. So there are, again, several different analytical attributes that one could evaluate with this. So thinking about cell therapies, again, things like transgene expression, endotoxin, microorganisms, presence or absence, looking for replication competent virus. There's a lot of analytical measurements that are required for these cell therapy products. And on the oligonucleotide side, of course, again, looking at things like identity, size and polydispersity of an encapsulated lipid nanoparticle therapeutic, or even the gene delivery vehicle composition, for example. So I think we sort of talked a little bit in high terms about what we're looking for here. We do certainly expect the performer to come in with an analytical target profile, an idea of the proposed analytical target metrics. So understanding what we're measuring, what types of limit of detection, limit of quantification, repeatability targets, and of course, ultimately reproducibility targets to ensure that these analytical methods are performing adequately. And I believe I'm also presenting topic number 4. Okay, topic number 4, wearable cell sorting and gene delivery systems. So as you can imagine, this proposal idea also comes from the thought about distributed manufacturing. And as many of you may know, the current embodiment of distributed manufacturing is actually, excuse my dog has decided to come in and say hello. The current embodiment of distributed manufacturing is acquiring cells from the patient. Those cells essentially get isolated, sent to a manufacturing facility that's very far away. The centralized manufacturing facility, for example, will transduce, in the case of CAR T, a T cell to have a new antigen expressed on the surface. It's then sent back to the patient where it provides its therapeutic efficacy. So you can imagine that all of this shipping back and forth of the cells and doing some of this biochemistry in the centralized platform, we'd like to move this much closer to the patient. So there has been development of these manufacturing systems that can actually be used closer to the patient. But what we'd like to do in this particular small business is start to evaluate the opportunity to even make that smaller and make it even closer to the patient and sort of take the manufacturing side out of some of this treatment. So what you can imagine is a miniaturized version of an apheresis device, something that can perform cell sorting to a sufficient degree that maybe we don't need a gene delivery system that can target that specific cell in vivo, but we're using sort of a mechanical cell sorting to get the cells pseudo-isolated quickly at the point of care. And then we can use some sort of mechanical device, whether that be sonication, a patch mechanism, maybe some sort of engraftment device to actually enable delivery of the gene directly as those cells are being extracted from the patient. So we're doing all of this then essentially in a closed system that's at the point of care in the hospital room without having to send samples even to an on-site manufacturing facility, but trying to do some of this gene delivery system right at the point of care. So in this paradigm, again, these cells are going to have to be sufficiently isolated. So there will have to be a degree of evaluation that the specific cell type that's going to be targeted is being sufficiently isolated by the device, whether this is a microfluidic device, perhaps it uses spiral cell sorting. There's a number of new technologies that have come out in that area that we can, of course, have to determine that we're actually getting sufficient resolution of those cells. For the second area of this, the ex vivo gene delivery, again, we're trying to do this at the point of care. So trying to not have to do a lot of cell expansion, not have to do a lot of cell expansion or cell activation, but trying to do this directly from cells that are basically taken out of the patient. So again, this would be a device that's very close to the patient, very easy to use, and enables gene delivery to that system. So you can imagine in the future a combination where each of these two sort of work in tandem. The cells are taken out of the patient, isolated, and immediately that gene delivery system is added to the cellular milieu to add transduction, and the cells could go directly into the patient. So this is a very early stage type technology. Nothing like this quite exists yet. So we're looking for these new business innovation ideas that can fabricate these particular devices, demonstrate that the target cells can actually be separated. Again, this wouldn't be using a patient necessarily, but using a complex media like, let's say, blood or serum. And then evaluating that we are actually able to then incorporate genetic material into those cells. Again, this could be done offline with traditional analytical measurements, but we're really showing proof of concept that we're isolating cells and then delivering those genes to the correct target cells in this sort of offline fashion. And last but not least, the phase 2 activities. Again, we'll do this in a more representative system as necessary. But again, demonstrating the performance of these systems with offline methods. And as I'd mentioned, both with this and the analytical, it's highly dependent on sort of the embodiment of the proposal. So what the gene delivery system is, what the cell type is. So some of these metrics are going to be a little bit squishy as written on the slide, but we're really looking for performers to tell us what they believe the degree of possibility is with their technology. So with that, I will turn it over to Adam Becker, the other program manager in our division. Adam?
Thanks, John. Hi. Can you hear me okay?
Yes, we can hear you.
Great. So I'm Adam Becker, Senior Advisor for Program Management at the Scalable Solutions Office. So this topic, if next slide, please. Slide 8, there you go. So this topic really, falls in line with our strategy to extend the extend the reach of healthcare, and by resolving barriers to access that result in health disparities, just whether it's from geography, demographics, or socioeconomic status. And so part of extending the reach is to really look at the home environment and say, how can we fully leverage the home environment and kind of common commercial devices, as well as wearable devices in that kind of healthcare experience, and to ensure that everyone can benefit equally from the wearable technologies revolution to optimize their health. To kind of meet that goal, of course, the wearable technologies need to be more affordable, but they also need to have sufficient data quality to be really fit for purpose for health applications, both currently non-regulated wellness applications and regulated health applications, as opposed to purely the kind of the fitness market. So in this topic, what we're this is a early phase. It's phase 1. So we're looking at TRL 2 through 4. It would be and just a proof of concept demonstration using breadboard would be completely fine, with supporting kind of analyses and projections on the miniaturization and cost and path forward from a licensing perspective and a regulatory perspective. And the initial focus on this was that blood pressure amongst the kind of 5 vital signs, heart rate, pulse ox, respiratory rate, skin temperature, was the blood pressure was not as equal quality, and not as well integrated into multifunctional devices. And although we're kind of initially looking at wearable devices, certainly ubiquitous devices like smartphones and tablets that are with the person all the time, could be an acceptable solution. I think there was a question about whether remote or contactless PPG, using imaging, would be acceptable in terms of solution. And I'll say yes to that, because it's a device that's in the home that everyone has, and if we can, as long as that platform can support kind of multimodal data to support multiple downstream health software applications, then I think it's a viable approach. Clarification on the slide where it says self-powered or USB-C. Well, USB-C, what I'm meaning by that is that it's rechargeable using a standard interface. Certainly preferably self-powered. And I think that is all I have to say on that. So I'm going to pass it off to Jenica. Thank you very much.
Thanks, Adam. I'm here to present topic 5, which is precision brain targeting, non-invasive delivery at the right place and time. This topic is very near and dear to my heart. I'm a neuroscientist by training, and I've been working in the neurotech field many years ago at DARPA. So we tried to identify unique ways to non-invasively target the brain. It's a hard problem, and it's a big blocker for a lot of neuroscience research, neurologists, and really ways to be able to appropriately treat neurodegenerative and psychiatric disorders. So we're hoping this topic is the first unlock to really be able to non-invasively target the brain at the right place and the right time. So I know there has been many strides to develop non-invasive delivery to the brain, but limitations definitely still exist. So there's some limitations include specificity, timing, control, and accessibility. And it really shows to reduce effective treatment and diagnostic tools for the brain. The current treatments usually involve invasive surgeries, maybe a direct injection to the peripheral or central nervous system, a systemic drug administration, which a lot of these can be imprecise, inefficient, imperfectly timed, and associated with undesirable side effects. So we want to change that. We want to try to be able to change the trajectory of non-invasive delivery. That was on the therapeutic side. Also on the diagnostic side, there's a large limitation, at least in terms of self-report and subjective tools, especially for neuropsychiatric disorders. Also, there's costly imaging techniques that have low resolution, and the timing might not be appropriate, especially over the course of days, where maybe imaging would be sufficient and helpful for diagnosing various psychiatric disorders. And we want to make sure that we can be able to diagnose early and accurately to make sure that we can bring in more effective treatment. So as many people know, brain disorders ranging from neurodegenerative diseases to psychiatric disorders are the leading cause of disability globally. And within America, 1 in 5 Americans live with mental health disorders, and over 1 in 3 are affected by neurological disorders. So we want to see if we can help prevent or help try to identify more effective treatments. So what we're looking for is to look at non-invasive ways and innovative approaches to target the brain in the diagnostic space, in the monitoring space, in the therapeutic space, or a combination of those 3 that don't require a direct injection into the central nervous system, the peripheral nervous system, or the cerebrospinal fluid. They need to show brain region as well as cell type specificity. We want to make sure there is a precision and timing in that potential diagnosis or therapeutic effect, as well as an on-off switch that is built in to maximize the therapeutic and diagnostic impact while minimizing off-target effects. So if, say, something is going wrong, we can turn it off. Some quick ideas could be maybe advancing imaging techniques beyond what's already available, beyond the MRIs, the PET scans, et cetera, nanotechnology-based delivery systems, novel small molecules, and viral-based innovations, something that's beyond the AAV vectors that we've seen that can cross the blood-brain barrier, however, have many off-target effects that can be easily translatable to humans. So there's been a lot of issues with developing a unique technology, but the translation just doesn't, it does not translate well to humans. So we want to try to reduce that barrier. So within the submission, the proposer should really articulate a proof of concept of what the delivery system is. I need to read it and say, what is this? So make sure you articulate exactly what that delivery system is, how it's going to precisely target the brain region, and the cell type of interest based on the indication that you're looking at. Understand how you're going to express that agent for a nominal amount of time, and what's the rationale for that time? How does it, for example, how does it correlate with a therapeutic effect, or maybe, a way to precisely diagnose a person? And how can that agent be precisely controlled? So what does that on-off switch look like? We also want to make sure that the proposer targets an inflection point in their proposal. So for example, maybe you have a device you're working on, you're ready to submit a direct to phase 2. We want to see by the end of direct to phase 2, you're going to submit an IND, or you're going to have an IND ready and approved by the end of phase 2. Or maybe you can do the first in-human demonstration as an example. What we're not looking for is direct injections into the central nervous system, the peripheral nervous system, or the cerebral spinal fluid. That is out of scope. And we do not want to fund anything that's incremental or what is already out there. If you're submitting a phase 1, I forgot to mention, we're looking both at SBIR and STTR, as well as phase 1 and phase 2. So in phase 1, we'd like to see a demonstrated proof of concept with a validated delivery method showing the right place, the right time, and a way to control the expression, that on-off switch, in vitro or in vivo. Phase 2, we want to see an in vivo demonstration of the proof of concept in animal models and an inflection point reach. So be that IND, IDE approval with the FDA as an example. We came with the problem, we want you to come with that solution. So you have to identify what is that appropriate inflection point? What are those appropriate metrics that you need to show to prove that technology? And then as well for phase 2, we want a confirmation of targeted delivery and a plan to integrate the technology with clinical practice or personalized medicine strategies. Next slide, that's for topic 5. I'm also going to present topic 6. So this is our food as medicine approach. So it's called Nutritech, Revolutionizing Personalized Food as Medicine. So as many people know, many Americans have a diet-related chronic health condition. 20% of young people aged 2 to 19 and 42% of adults suffer from obesity, which has an increase in risk for chronic health conditions. And this has drastic implications on health outcomes across our country. And there's a lot of things we still don't know to help with this, this particular indication for humans. So how do we adequately assess individuals' nutrition needs? And how do we precisely guide what we should eat? Current techniques are limited by subjective and outdated measurements, and also looking at a one-size-fits-all recommendation where maybe we should be focusing on these individual needs or a precision medicine type approach for food, really to improve health outcomes for all. So what the topic is really trying to meet this challenge. We want to have the proposers develop either a novel digital health and diagnostic tool that can inform precision medicine for nutrition. We really want to understand the intersection of biological, environmental, metabolomic, and social factors to help inform certain things, such as malnutrition, micronutrient deficiencies, or factors that lead to diet health related conditions, and have these specific precision nutritional interventions to improve health outcomes and inform food as medicine for everybody. The ultimate goal of this particular topic is to eventually have an FDA-approved diagnostic assay or a digital health tool for precision nutrition. So we want to make sure that this solution you're creating will be regulated by the FDA. What is out of scope are a one-size-fits-all approach. We're not looking for that silver bullet for all. We're looking for those individual approaches for everybody. And we are not looking for products that only inform the promotion of a healthy lifestyle. So general wellness products. And the topic includes what we're looking for is a demonstration of technology feasibility for the digital health tool or the diagnostic to evaluate specific nutritional needs of a patient, including the intersection of environmental and biological. So what I mean by biological is maybe looking at the intersection of genetics, epigenetics, gut microbiome, and endocrinology, and factors that impact metabolism, as well as combined social factors. So some examples there are social determinants of health, dietary habits, lifestyle habits, cultural connections to food, as well as other health data. And within your proposal, you really need to describe how you're going to reach the objectives of a proof of concept for demonstration, as well as the technical feasibility. I forgot to mention, we're only looking here for phase 1 proposals, and we're looking at SBIR and STTR. And just quickly for digital health within your proposal, make sure you describe how you're going to develop your technology platform. What is it? And how it can assess nutrition status for individuals of a particular population, and feasibility to provide a precision nutritional intervention. For diagnostics, we want to make sure that you describe to us in your proposal how you're fully going to develop that diagnostic assay. What's in it? What does it look like? How is it going to assess individual nutrition status and feasibility to correlate with a nutritional intervention? This includes completion of an analytic validation study, looking at sensitivity, specificity, accuracy, precision, and other relevant performance metrics. And we want to make sure they are above the state of art that is already out there. And lastly, for all of the solutions that are submitted to us, we are very interested in looking at what is that associated population that you're interested in targeting. How are you going to establish an engagement or a partnership with that population to make sure we're accelerating the transition of your particular solution to the hands of the right people that you want to work with. So that's all I have, and I'm going to pass this over to Jason Smith to talk about our last topic.
So much, Jenica. Hi, everyone. So Ileana is currently out on a very well-deserved vacation today. So I'm Jason Smith. I'm one of her team CETAs, and I'll be talking about topic 7, clinic-ready imaging devices and protocols for visualization of the inner ear with high accuracy. Let's get into it. The senses of hearing and balance are vital to daily human functioning. Understanding the root causes of hearing loss and balance dysfunction are oftentimes dependent on the ability to closely examine the anatomy and physiology of the inner ear. At the moment, clinicians do not have a way to image the inner ear with sufficient fidelity. To address this challenging clinical problem, this announcement requests the development of novel, non-destructive technologies to image the anatomy and function of the inner ear with high detail in human patients. For this, we are accepting direct to phase 2 and fast track proposals only. And potential areas of focus include are all listed here with this green check mark. So discerning bone from surrounding media in the inner ear, detailed assessment of inner ear physiology, as well as capturing stereocilia motion and other dynamic movements, and the identification and localization of otoconia in the vestibular system. And with respect to what will not be considered for funding, as we emphasized in the title, this funding call is not meant to be interpreted as a research opportunity. A clinic-ready device and imaging protocol are expected by the program's end. With respect to key outcomes and milestones, they are listed in part here. By the end of phase 1, it is expected that the performers will achieve non-destructive imaging of the cochlea and or vestibular end organ in less than 1 hour. Though non-invasive approaches will be preferred, invasive approaches, such as those that enter the round window, will also be considered. And depending on which way your team decides to go, the expected resolution for invasive approaches will be significantly higher, which should reach at least 2 cubic microns than non-invasive approaches, which should reach at least 100 cubic micron. By the end of phase 2, the technology developed in phase 1 will undergo safety evaluation in large animal models, such as pigs. Then the validation of the device in awake human volunteers and patients suffering from hearing loss will play a large role in preparation of a 510(k) submission. Just keep in mind there are many more milestones listed on the solicitation document. So if your team is interested in submitting, please make sure to go over to the topic 7 section of the solicitation, which can be found on sam.gov. Thanks for listening, and please feel free to reach out to our dedicated SBIR email account with any questions that you may have. Up next, we have Alexandra, who will give us an overview of the submission process. Thank you.
Hello, everyone. I'm Alexandra Rouse. I'm with GSA, and I get to talk about acquisition after those fun topics. We are so excited to be an interagency partner with ARPA to support their mission. We are conducting the contracting support on the solicitation to help ARPA-H meet its mission and to move forward public health. So it's an exciting project that I know all of us here at GSA are super excited to be a part of. And if you don't know why after those last descriptions, I don't know what to tell you. But because I am discussing acquisition, I am going to remain very close to the messaging that are on the next several slides. At this particular point, though, I would like to go rogue and share some best practices for industry as you're thinking about preparing an offer for this solicitation. Probably the most critical and most obvious is to read the solicitation, deliberately read the solicitation to understand what it is that ARPA is looking for and what's required as far as your submittals for us to evaluate and ultimately get you to award. make notes as you're reading the solicitation. And if you have questions or something there in the solicitation isn't clear or you don't understand it, please send us an email. The best way to get a hold of us on this particular solicitation is through a special email that we've generated for this project. It's GSA-ARPA-H-SBIR@gsa.gov. Again, GSA-ARPA-H-SBIR@gsa.gov. It's a mouthful. As I go through the presentation, I'll just say please send to the GSA email. Our contracting officers are Suzanne Kixmiller and supported by Cynthia Weaver in the contract specialist role. Another reason that this email box is so important is that we have released the solicitation, which means we are now in a controlled communications environment. Whereas before we could, folks within ARPA, folks within GSA could talk about requirements, but now with the solicitation being on the street, the only way to communicate with us is through that email box. If you reach out to anyone else on this call or any of the presenters that you've seen today, they'll politely direct you to this email box. So this will be your best tool during this phase of the acquisition. Next slide. So proposal submission. As was shared earlier, proposals are going to be should be submitted by July 26, 2024 by 11 a.m. Eastern Time. Again, we're sending everything to the GSA ARPA-H SBIR GSA.gov email box. And we are requesting very specific nomenclature for the submissions. So as you're writing your questions or submitting your proposals, please make sure you include your company name, the solicitation number, which is that fancy number that you see in bullet 2, 47 QFCA 24 R00036, proposal topic, and include the number of the topic that you are interested in. We encourage encryption for the proposal submission. Make sure you test the password protected documents before you send it. And you're also responsible to make sure that the separate email is submitted with the passcode so we can download the documents. And that would need to be sent under a separate correspondence from your submission. GSA, our email system runs on Google. So there is a limitation to the size of the file that you can send. We're limited to 25 megabytes with individual emails. So please be cognizant of that. If you send something that is larger, we will not receive it. If we don't receive it, we can't evaluate it. Okay, next slide. The offer eligibility and performance requirements. This gets into the nitty-gritty, basically ensuring small businesses are small businesses. We are asking that companies to secure a phase 1 or a phase 2 award, you have to qualify as a small business concern at the time of award. The eligibility guidance for the business sizes for SBIR are located on SBI's SBIR website, which is www.sbir.gov/about. And that gets into the specifics of what would constitute your small business concern. Each offer has to qualify as a small business for research or research and development purposes. And this certification has to be on your cover sheet of the business proposal submissions. Appendix B for phase 1 business proposal template or phase 2 would be Appendix D. If you're, well, I'll get to it in a minute. The awardees have to submit a certification that states that it meets the size, ownership, and other requirements of the SBIR program at the time of award, or any other time that's set forth in the SBA regulations. And we are also asking for both phase 1 and phase 2 research and research development work. It has to be performed by small business concerns. And if you're using subcontractors, they have to be located within the United States. If there's a situation where you have a unique circumstance, then send an email explaining the circumstance, and we'll explore it further, but the explanation needs to be sent to our SBIR, our GSA ARPA's email address, so we can proceed the evaluation on the special and unique circumstances. Next slide. Okay. So phase 1 and phase 2, there are similarities, but in the architecture of the solicitation, you need to be cognizant of which appendix that you're looking at. For the phase 1 proposal, the technical proposal is located under Appendix A. The business proposal is located under Appendix B. For the phase 2 proposal, there are nuances that are different. It's a little more mature than the phase 1. So the technical proposal there in this solicitation is listed under Appendix C, whereas the business proposal is listed under Appendix D. Both have appendices for A1, in Appendix A1, that deals with human subjects and clinical trials information form. If that's applicable to you, you will need to provide that. Under Appendix A2, there's a study record and an attachment to human subjects and clinical trials information form. Again, if that applies to you, then those need to be addressed as well. If you are considering a fast track submission, please be aware that 2 proposals will need to be submitted. For the phase 1, the concept, and the phase 2, which is the R&D commercialization support. Next slide. The evaluation process, basically, it goes through 2 reviews. The initial review is a preliminary review process, and we're looking at proposals which are deemed to have the highest non-price technical criteria, which is outlined in section 4.2 of the solicitation. It will be discussed and assigned an overall impact or preliminary confidence rating. The initial review is not applicable to the fast track proposals. Since the fast track involves a phase 1 and phase 2 submission, the initial review will be combined to address both phases. The final review, which would be the second part of the evaluation process, is having the proposals evaluated based on their individual merits. So, and the awards will be made to offers whose proposals are seen as the most advantageous to the government and are consistent with the instructions outlined in 4.2. And naturally, with all government procurement, SBIR or not, it's also subject to the availability of funding. Next slide, please. Okay. So pricing factors. We like price. We like to evaluate price. We always are interested in ensuring that the government and ultimately the taxpayer is paying a price that is fair and reasonable. We have different methodologies and access to a different evaluation techniques to assess the fair and reasonableness of pricing. If we find that a proposal is not reasonable in its pricing, it could result in a offer not being selected for award. And if the proposed pricing is higher than the published award ceilings for each phase, that would negate applicability for an award. So as Jenica said earlier, they got a waiver to raise thresholds for the phase 1 and phase 2s. So the dollar threshold or ceiling for pricing on phase 1 is $600,000, and the duration is 6 months to 2 years, whereas phase 2 and direct to phase 2 can receive, or the ceiling, the price ceiling is $3.5 million from 1 to 3 years. Next slide. Non-price factors. We consider non-price factors because that goes to the merit of the proposed approach that is given. So we want to make sure that the technical merit and the offer is sound. We want to see identification of clear and measurable goals, and we want to ensure that there's a reasonable chance of these offers meeting the topic objectives. There's also potential of proposed research for technical innovation. If the offer offers significant advantages over existing approaches, methodologies, instrumentation, or interventions that are currently used in research or in clinical practice. We're also interested in the potential of the proposed research for commercial application, which really goes to the intent of SBIR. We want to make sure that an activity could likely lead to a marketable product or process, considering the offerer's proposed methods for overcoming possible barriers to entry in the competitive market landscape. And we're also looking at the qualifications of proposed principal investigators, project directors, supporting staff, and consultants, and the appropriateness of the leadership approach that is described within the proposals. Next slide. So our technical evaluation criteria is really based on high confidence, some confidence, and low confidence. We will assign the level of confidence that we see as part of our technical evaluation. This is considered equal importance, and confidence ratings will be assigned using these three measures. The high confidence, it describes that the offerer understands the requirement, proposes a sound approach, and will be successful in performing the contract with little or no government intervention. Some confidence, again, it's very similar to high in that some confidence that the offerer understands the requirement and proposes a sound approach and will be successful in performing the contract with some government intervention. And then low confidence, government has low confidence that the offerer understands the requirement and proposes a sound approach, or will be successful in performing the contract even with government intervention. So these are the things that you should be thinking about as you're preparing your offer and how they can be interpreted by the evaluation panel. Next slide, please. Okay, award decisions. Okay, to receive an award, the offerers must be eligible according to the requirements defined in the solicitation. Again, we covered a high level of requirements, but not all the requirements in this presentation. But the government will make awards to offerers who provide the best overall value to the government, with the main considerations including ratings resulting from the technical evaluation, areas of high program relevance to the ARPA-H mission, to accelerate better health outcomes for everyone by supporting the development of high-impact solutions to society's most challenging health problems. The program balance, so balance among the areas of research that are tied to the solicitation, availability of funds, the price reasonableness, and the security risk that is assessed by the HHS due diligence program. Next slide, please. Okay. I forgot to mention that we're only awarding firm fixed price contracts with this solicitation. Grants are not going to be a part of the solicitation. Firm fixed price contracts. The solicitation questions are to be submitted in writing to the GSA-ARPA-H-SBIR@gsa.gov email box, and they are due by 11 a.m. on July 15th. Please be careful, please be aware and leverage the naming convention described here in the second bullet, where you have in the subject line, you have your company name, the solicitation number, the topic number, and indicate questions. We will use your questions sorry, I'm getting tongue-tied. We will use your questions to make sure that we are creating a solicitation that is in the best interest of the government. And there is a possibility that we would issue an amendment to this solicitation that will address responses to the questions that are submitted. We anticipate that responses would be published or will be published in a sufficient time for interested offers to consider them prior to the submission of a proposal. So, in other words, we'd like to have your questions no later than July 15th. We will go through, we will address the questions, provide responses, and if the amendments and changes need to be made, you will have anyone who is interested in providing an offer will have enough time to address updates should they occur by the closing date of the solicitation on July 26th. Okay. And that is my brief summation of the solicitation. Thank you, ARPA, for having us GSA participate. And now I would like to turn the floor over back to ARPA to Michelle Matrone.
Hello, everybody. I'm Michelle. I'm a program analyst at ARPA-H, and I help run the small business program with Jenica. I touch all parts of the program. I work with the acquisition office, GSA, also with our program managers. And I'm also a contracting officer representative on many of the contracts that we have already awarded. So I have the pleasure of working directly with our performers. Before I go over common questions that we receive, I wanted to note that we're not going to be doing a live Q&A. As we've received an overwhelming response of attendees to this webinar, which we're very, very happy about. And as Alexandra mentioned, so we're asking if you have any questions that have not been addressed during this webinar, please submit them to the mailbox identified in the solicitation, and we will be, or GSA will be posting that amendment on sam.gov. And lastly, just a couple tips for when you do submit your questions. Instead of submitting broad questions such as, does my technology fit within a certain topic, we ask that you please be very specific for those scope related questions. And we'll make sure that the program manager of that topic answers it. So just a couple questions that we get. So first one here is, I'm an academic scientist that is interested in funding this opportunity, but I don't have a company. Can I submit a proposal? So for all of our SBIR, STTR solicitations, you have to be an eligible US small business. And so you can just check out the general definition section of our solicitation that defines a small business concern and eligibility. Second one that we get is, my small business is interested in submitting a proposal. Can we apply as an STTR instead of an SBIR? So for this one, I want to note that this is our third solicitation, but is our first one where we're actually including STTR topics. But for each of the topics, read through, and in the first like couple sentences of the topic, you'll see whether it's an SBIR or an STTR, and then what mechanism it will be. Our PMs and teams, they went through all of the topics and they mentioned all of that, but it's also listed in our solicitation. Another question is, I understand that under the SBIR ST2 program, there's a requirement that work needs to be performed in the US. However, we wish to include one of our foreign collaborators. Is this possible? So foreign involvement will be considered on a case-by-case basis and must be justified very thoroughly in your proposal. And then note that small businesses and other partner organizations listed in the proposal are also part of the foreign risk assessment that takes place before the award can be made. And that's also one of the new criteria added to when an award decision is made, is the results of that foreign assessment. So definitely pay attention to that, and then also pay attention to the foreign disclosure form that's required to be submitted as part of your proposal. Next question is, will the same topics be listed in a later solicitation so that I can apply for an ARPA-H opportunity in that category once I have started my company? And then I'm also planning for an SBIR grant in September. Am I allowed to apply for and be awarded both the grant and the ARPA-H grant? So we encourage it. We encourage companies. You're welcome to apply for opportunities outside of ARPA-H as you see fit. But if you're applying within ARPA-H, we just ask that the applications do not overlap in content. And then lastly, is it permissible to have 2 different PIs for the phase 1 and phase 2 portions for the fast tracks? And the answer to that is yes. So now I'm going to pass it back over to Jenica. She will have closing remarks.
Thanks everyone for sticking around. I know that was a lot of information, so please make sure to refer back to the webinar recording. Also, please take a look at our website. Here is a QR code on the left-hand side to learn more about our small business program. And also look for more opportunities. Maybe this solicitation isn't right for you, or it is, but you have another idea. Take a look at subscribing to our ARPA-H Vitals. It's our newsletter that comes out, I believe, once a week. So you learn about all the unique opportunities on how to work with ARPA-H and our different funding mechanisms. So with that, thank you so much for your time. And we look forward to reviewing your proposals in the future. Take care.